Students. So let's go through the levels of protein structure really quickly because I got a lot of questions about it in office hours this week. Okay, so this website that I'm showing you right here, this is BioNinja. Fantastic website if you like reviewing by like reading or looking at pictures. It's one of my favorites, and I include links to this in the class links page on OneNote a lot. Okay, so let's talk about primary structure. So primary protein structure is just the order of amino acids, so like a string of amino acids all in a chain. And so we could imagine, I'm going to make myself a little bigger here. So you can imagine it might look something like this. So this purple rod represents the covalent bonds, the peptide bonds is what they're called, between amino acids. So like here would be one amino acid, and here is another, and this purple would represent those covalent bonds. So primary structure is literally just this string of amino acids all in a big chain. That's it. That's it. So different proteins will have different primary structures if they have different amino acids here. So like if I switched out this amino acid for this one, then I've changed the primary structure. Okay, so that's primary structure. What about secondary structure? So secondary structure is we take this little chain and we form two really common shapes. And because these shapes are so common in proteins, they have special names. So the first one is this curlicue shape. So it's going to look something, if I use my little rods here, it's going to look something like this. And these curlicues are called alpha helices. A helix, helice is the plural of helix, and a helix is just a spiral. Okay, so that's the first shape. The second shape is this one here, which looks sort of more like a zigzag. So it's going to be something kind of like this. This is called a beta pleated sheet, or sometimes I just say a beta sheet. Okay, so those are the two primary, or the two secondary protein shapes, beta sheets and alpha helices. And a protein can have both. It can have more than one of each. So I'm going to add another little alpha helix over here. Okay, and these secondary structures are held together with hydrogen bonds. Hydrogen bonds are what hold together these secondary structures. Okay, and so all we did for secondary structure was we folded up the primary structure, right? So we're going to follow that same pattern when we get to tertiary structure. For tertiary structure, we're just going to take this whole thing and fold it up even more. So maybe it kind of like crunches in, something like this. So this is sort of the final three-dimensional shape. This is tertiary structure. So how does it know what shape to make? Well, that's going to depend on the amino acids that are in the protein and the bonds that they can form. So in my little example protein, see how like this amino acid is kind of tucked into the inside of the protein? That's probably because this amino acid is hydrophobic. So it's hiding from all the water that is all around this protein, dissolving it. So things like the polarity of the R groups are going to dictate how it's going to fold in tertiary structure. The types of bonds that hold this together are all the types of bonds that can form between R groups. So it's going to be hydrophobic interactions, ionic bonds, hydrogen bonds, and one very special type of covalent bond, which is this one right here, which is called a disulfide bridge, that can only form between these two amino acids that have sulfur in them. Okay, so that's tertiary structure. So then what is quaternary structure? So if we have one chain that has its own three-dimensional shape, and then we have another completely independent polypeptide chain, so different amino acids all joined together. If these two have to work together to be a functional unit, that's called quaternary structure. So quaternary structure is just one or more of these polypeptide chains, and they're held together by the same types of bonds that tertiary structure is. So they're held together by hydrogen bonds, covalent bonds, ionic bonds, and hydrophobic interactions. Okay, so those are those different types of structure there. Okay. So that brings us to sort of the next thing. So here's our finished protein. It's got its primary, secondary, and tertiary structure. So let's talk about denaturation. So all that denaturation is we're going to lose all of the structure except for primary. So here's a nice picture showing what that looks like. So we can imagine, I'm going to do this with the green one because it's got less stuff on it. Okay, let's imagine here is our protein. It's got its primary, secondary, and tertiary structure. This protein happens to not have quaternary. So how does this protein become denatured? Well, in order to denature it, we are going to need to break the bonds that hold together tertiary and secondary structure. So in other words, we're just going to go like that, and you'll get something like this. So it's important to understand that during denaturation, you do not break those covalent bonds that make a primary structure. So I can't break that, right? So we still have primary structure, but we lost everything else. Okay, so let's talk about how that works. So let's use as our first example, heat. So as we increase the heat of something, we're just going to increase the movement of molecules, right? So heat is just kinetic, or heat's going to increase the kinetic energy. So as you raise the heat, this molecule is going to like vibrate more and more, right? And we can imagine if we raise the heat enough, I'm going to hit myself when I do this, that this is actually going to vibrate so hard that it's going to sort of like vibrate itself apart. And these molecules will start to move so quickly and vibrate so much that they will break those bonds that hold together secondary and tertiary structure. Once again, they have not broken the covalent bonds that form primary structure. So that is how heat denatures proteins. It literally just makes the molecules move so quickly that they shake themselves apart from those hydrogen bonds and ionic bonds, hydrophobic interactions, things like that. It's pretty much everything but a covalent bond. Strongest type of bond, right? Okay, so how about pH? So pH, what pH is it's just a measure of the amount of hydrogen ions in a solution. So for example, something that's really acidic has a really high pH, so it's got a lot of ions. So let's imagine, here's our protein. We've got all these little R groups in here, right? Let's imagine that we've increased the pH, so it's more acidic, or sorry, we've decreased the pH, we've made it more acidic. So now there's a bunch of hydrogen ions floating around, and they're going to bind to some of these R groups and change the charge on those R groups. Well, if we change the charge on those R groups, maybe, here there used to be an ionic bond between these two R groups. Let's say this one up here used to be negatively charged, but because there's so many hydrogens, the hydrogen pops on there and the negative charge goes away. That's going to, break that ionic bond. So now the protein's starting to unfold. And because this is less stable, it's going to break other bonds. And so it's just sort of like a chain reaction. And then end result is we get the same thing, which is the protein becomes denatured, which means it loses quaternary, tertiary, secondary structure. Primary structure is still intact. Okay. So yeah, that is in a nutshell what you need to know about protein structure and how proteins denature. I recommend you check out, this is question 30 in your study guide. This is a summary of protein structure, the different levels and the different bonds that hold together those levels, and some very beautiful pictures for you. So it is crucial that you understand this stuff because proteins are important, right? Okay, that's it. Good job.